Today's topic is going to be focused on PacBio HiFi sequencing and how it can help you overcome some common challenges in microbiome research, including ambiguous taxonomic assignments and culture-resistant bacteria. It's my pleasure to introduce today's speakers who will be lending us their expertise. The first will be Bing Ma from the University of Maryland School of Medicine, and the second will be George Weinstock from the Jackson Lab. So we have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a Q&A session. You're welcome to submit your questions at any point in the webinar by typing them into the question tab on your dashboard. We'll be recording the webinar and making it available for download in the next few days. I also wanted to point out that we have provided with you with 3 useful sequencing handouts that you can download also from your attendee dashboard. So with that, let me get started by giving you a brief update on PacBio's metagenomic solutions for the Sequel II system. So all of the solutions that we'll be talking to you about use HiFi sequencing. So HiFi is a unique technology to PacBio, which allows us to sequence many, many times around the same SMRT bell and then use each of those subreads to do intramolecular error correction to generate one consensus HiFi read that is both very long and very accurate. So the way we use this in metagenomic sequencing is through 2 different applications. The first is 16S sequencing. And on the Sequel II system, you can expect to get up to 3.6 million full-length 16S sequences, which will let you multiplex at either 96 or 192 plex, and to get over 10,000 reads per sample. We also offer you shotgun sequencing solution. And with this solution, you can get up to 2.4 million reads, each of which is about 10 kb in length. For 16S sequencing, we have a couple of different ways that you can use this application. The first is by using our own newly updated 16S protocol, which uses an asymmetric barcoded system in order to do very high levels of multiplex. And this is quite affordable, and you can find service at all of the usual places that you would go to get PacBio sequencing done. You can also use an all-in-one kit provided by our partners at Shoreline Biome, and this kit allows you to do DNA extraction, amplification, and also analysis all in one solution, and provides you access to their curated database of full-length 16S sequences. For metagenome sequencing, the advantage of PacBio HiFi is that gene prediction can be done directly on the HiFi reads without needing to do any assembly at all. And this is an advantage because, as you probably know, typically when you assemble short read data from shotgun sequencing, anywhere between 20 to 70% of that data will not map to the resulting assembly, and therefore that information is not accessible for things like gene finding. With HiFi sequencing, every single read yields 7 to 9 completely intact genes, and this lets you get information even from species that have very low representation in your data, because you don't have to have enough coverage to do either assembly to improve the contiguity or to do error correction in the case of other long read technologies. And even better, the high accuracy of HiFi reads means that you can use the existing NGS tools and pipelines that you're already familiar with without any modification. Okay, so now I would like to introduce our first speaker, Bing Ma. So Bing is a research associate at the Institute for Genome Sciences on the Department of Microbiology and Immunology at the University School of Medicine. She received her PhD from the University of Wisconsin with a focus on genome sciences. She went on to do a postdoc with Jacques Ravel at the IGS, where she studied how the vaginal microbiome impacts susceptibility to infectious disease. She's now on the IGS faculty, and she's here today to talk to us about her work on the gut microbiome of premature infants. Welcome, Bing.
So in today's talk, I'll focus on the population of early preterm infants and the methods we use to study their gut microbiome. As we know, preterms are especially vulnerable. Many requires intensive care just to remain alive. In US, the preterm rate is about 10%, and worldwide it's about 11%. So that translates to 450,000 in US per year and 50 million worldwide a year. And based on their gestational age, preterm births can be characterized to either moderate or late preterm if they were born after 33 weeks of gestation, or extremely preterm or very preterm if they're born before 37 weeks of gestation. And and the early preterm contributed majority of the fatality rate of neonates, and the leading cause of neonatal deaths are prematurity, pulmonary conditions, and necrotizing enterocolitis or NEC. The first two happens within the 24 hours after birth, and NEC happens about 2 to 4 weeks after birth. And NEC is a severe bacterial infection that happens 2 to 4 weeks that affect about 10% of early preterm births with a mortality rate about 50%. It happens when bacteria normally that are normally confined to the intestinal lumen that translocate across impaired epithelial barrier and triggering a cascading inflammatory response. And ultimately, it leads to necrosis, perforation, and systematic infection and inflammation, and potentially multi-organ failures and death. And intestinal immaturity is a proximate cause of susceptibility to NEC. The matured intestinal barrier is lined with a continuous monolayers of epithelial cells populated by community of bacteria. And these bacteria are considered to educate immune system and regulate the expression of the tight junction proteins in between epithelial cells to form a tight but selective barrier to prevent the bacteria to translocate across the luminal side. While in the immature intestinal barrier, because of decreased mucus and decreased IgAs, and the low intercellular junction integrity, and all of which are attributed to either host factors or altered microbiota. Some say it's caused by antibiotics, and all of which has led to increased permeability, and then leads to bacterial translocation, that leads to exaggerated inflammation, tissue injuries, that eventually leads to NEC. Apparently, to promote intestinal barrier maturation to prevent the bacterial translocation will help to reduce NEC incidence. So in this study, we target intestinal permeability, and there are many outstanding questions remains. So our goal is to determine the window period to monitor intestinal permeability. We want to characterize gut microbiome and their early establishment in intestinal barrier maturations in early preterms, and want to know what factors affect the establishment of the leaky gut associated microbiota. The golden standard currently used to access gut permeability is through measuring the double sugars. That's non-metabolized sugar probes, lactose and rhamnose, that measures the sugar goes in between the cells. That's a paracellular uptake markers, or goes through the cells. That's a transcellular uptake. And their ratios indicating how leaky the gut is. So in our first study, our goal is to enroll 50 subjects. These are preemies, were born less than 33 weeks of gestation, and we recruit them within 4 days of age. And at 3 time points, we measure their gut permeability right after birth, at end of 1 week, at end of 2nd week. And same time, we collect their fecal samples. We sequence their fecal microbiota using the short regions of B3, B4 of the 6S on MySeq. And at the same time, we collected neonatal factors and all of the other feeding practices, other factors that may potentially affect the microbiota. And the first thing we see is there's a rapid decrease in intestinal permeability over the first 2 weeks, especially between the first day, right after birth, and at end of 1 week, there's a sharp decrease. And if we lay over the intestinal permeability with microbial diversity, there's a steady increase in their biodiversity, and that diversity increase correlated with a decrease in intestinal permeability. And if we stratify their gut microbiota by different time points, that's right after birth, at the end of 1 week and 2 weeks, and compare high and low IP subjects, and using a statistical models we use what we call zero inflated negative binomial random effect models. We reviewed Clostridialis is significantly more abundant in infant with low IP. And also we observed Clostridialis is also associated with early breast milk feeding and less antibiotics exposure. So this is very exciting results. However, their limitations and outstanding questions remain. First of all, it's a pilot study with a small cohort. And second, the taxonomic resolution using the short regions of 16S is not optimal. And also, we did not observe too much or very low abundance of bifidobacterium, which is not consistent with previous study that in breast milk fed neonates, there are more bifidobacterium. So with that question in mind, we designed our second study. So in the study, our goal is to expand the cohort size to 200, and we collect daily samples from this subject, and we measure their intestinal permeability at the end of 1 week. And when we wanted to improve the taxonomic resolution, so that's why we sequenced both their V3, V4 regions using MySeq and also the full-length 16S on SeqO2. And we also pick some samples and sequence their metagenomes using both short and long read sequencing. And the rationale of using full-length 16S to improve the taxonomic resolution is because their previous studies, they report there's a low concordance among the different 16S regions. We use the short amplicon to characterize the taxonomies. There isn't much concordance between the amongst different regions. And also they're using the V4 regions, which is what most of the gut microbiota study uses, and their poor taxonomic accuracy. In our first study, we used V3, V4 regions, but still the taxonomic resolution is not very optimal. So we went ahead using both short and long reads to sequence the 16S. And first thing we observed, there's the qualities across the cycles for the long read sequencing. We did not observe quality deterioration towards the end comparing to using short read sequencing. And it's actually common to see towards the end, there's a quality drop. So we have to do rigorous quality trimming. For the long read sequencing, we got about a little over 7,000 reads per sample. And the average length is 62 base pairs, more or less, after we remove the primers. And for the short read sequencing, the sequencing depth is much higher. The length is about is a little over 400 base pairs. So using the full length 16S sequencing, we generated 1376 ASVs amplicon sequence variants using data2. So there's a data2 protocol recently published for the full length 16S protocols. For these, majority of this 13,000 ASVs, that's about 87%, are able to assign down to species level. And that's very exciting. For the rest of them, for about 171 ASVs, we cannot assign to species level. That's mostly due to the lack of 16S ribosome RNA references. So if we pull up all 1,376 ASVs, we constructed this phylogenies, and what this shows is each of the tips of this tree is ASVs, and each of the dot, because it's longitudinal study, so this is at a different time points. So if we have done 16S based microbiota studies, you see it's, especially for the fecal microbiota, it's not very usual to have a phylogeny, 16S phylogeny with resolution like this. And we also in our sequence, we use Zymo Biomics microbial community standard as internal control to get an idea of the compositional biases. So this one in the middle is theoretical compositions for Zymo Biomics, and this is the full-length sequencing. So if you, this one is quite similar to the Zymo standard, and this one's a little deviated. It's probably because the sequencing depth for the sample happened to be not very. It's about like 2,000 reads per sample. But compared to the short read sequencing using the short regions, the composition is very different. And the taxonomic level is also, some of them are at a genus level. And it's overrepresentation of particularly Salmonella and E. coli, and underrepresentation of other groups. And in the studies, we particularly focused on Bifidobacterium because we wanted to characterize its abundance in this cohort. And what it shows here is the short read sequencing in terms of the percentage Bifidobacterium is actually is significantly different using the full-length 16S sequencing. And also in terms of prevalence, this means in many of the samples that have low abundance of Bifidobacterium, but in many of the samples, while in the full-length 16S sequencing, there's only fractions of sample, but if they're present, they're in a higher abundance. So we also use the WGS metagenomic short read sequencing of the samples, which shows abundance-wise, it's similar to the full-length 16S sequencing, since also the prevalence is similar. So let me show you what it means. So for each of the sample, each of the bars is the samples. So this is the full-length 16S sequencing. This is the short region. This blue area is actinobacteria, which is Bifidobacterium. So you can see it's a lot for these samples with high abundance of Bifidobacterium. The short read sequencing, they don't have as much. They don't have as abundance as these samples, but in lots of the samples, they have a lower abundance, but in many of the samples. So from comparison, you can see the short read amplicon sequencing, they underestimate the abundance of Bifidobacterium, but overestimate the prevalence of Bifidobacterium. So coming back to our study, we stratify this subject by their post-menstrual age and compare high and low IP. We observed Clostridia, where Clostridiales is actually more abundant in low IP compared to high IP. This result is consistent to the first study. And on top of that, we observed actinobacteria, that's actually Bifidobacterium. It's more abundant in low IP than high IP. And this is not observed in the first study. And it only shows in the second stages study. We went ahead and checked all the factors, or variables in the context of IP. We find two groups of factors. One group is the birth weight, gestational age, or postmenstrual age. These factors are collinear or host factors. They are significantly associated with high IP, which means if the infant were born in early gestational age or low birth weight, they're likely to have high IP than low IP. And the other group of factors are rather modifiable factors comparing to this birth weight or gestational age. These are the durations the infant received mom's breast milk feeding, or the duration of antibiotics uses. And these factors also find associated with IP. So if we factor in those different factors in the context of microbiome, what we observed is the Clostridiales and Bifidobacterium, they're more abundant in a low IP subject compared to the high IP. And what this result tells us is these two groups of bacteria, they're associated with low IP. They're also more abundant in a subject that has longer days of mom's breast milk feeding and less days of antibiotics treatment. It is possible that these two factors promote these two groups of bacteria to grow, and these bacteria can in turn affect intestinal barrier maturation. So another question we want to know is what exactly the species in the Bifidobacterium involved in low IP. So we did a average nucleotide identity clustering of all the ASVs that were taxonomically assigned to Bifidobacterium, and we reviewed as different subspecies for that species. There are actually 19 subspecies for Bifidobacter longum, and 2 of which are most abundant in this cohort. And also 5 subspecies of Bifidobacter breve, and 1 of which is very abundant. We also reviewed a lot other subspecies from Clostridiales and Lactobacillus. So if we pull up all those bifidobacteria ESVs, the full N16S, and constructed a phylogeny and colored them by different subjects, what we see is there are multiple subspecies of bifidobacterium can exist in one cohort, for example, Bifidobacter longum, or a single subspecies of Bifidobacterium can share in between multiple subjects at different time points. So this result indicate a high microbial community dynamics of Bifidobacterium and a rather strong environmental adaptive advantages in infant gut. And then we take some of the samples, we sequence their metagenome using long read sequencing on SQL2. And we pull up what we do is we pull up the samples with the DNA already extracted using our routine DNA extraction kit that involves the bead speeding step. We do not re-extract the samples, use the long molecular weight protocols, simply because we don't have that material. So we pick the samples with around 500 nanogram of DNA, more or less, and we sequence them on SQL2 and sample them using Canoe, and we bin them and constructed a metagenomic assembled genomes. And what this table shows is all this close circularized metagenomic derived genomes assembled from the assembly results, and one of which being a closed genome of Bifidobacter breve from the bin metagenomics results. And after annotation, we reviewed a efficient carbohydrate utilization pathway, or BIFISHUNT. This pathway is not, it's only existed in very few Bifidobacterium, and only for these bacteria that are called champion colonizer indicate this genetic capabilities of metabolizing human milk oligosaccharide to short-chain fatty acid, particularly to acetate. And acetate is actually a required substrate for Clostridiales and other fermentators to grow on and to generate butyrate. And butyrate was known to have the benefit of promoting intestinal barrier integrity. So in summary, in the study, we characterized the gut microbiome in a high resolution to study intestinal barrier maturations in early preterm neonates. And we find there are multiple factors like gestational age, breast milk feeding, and antibiotic exposure associated with low IP. And also, we find the 2 biomarker groups, Bifidobacter Clostridiales, are associated with low IP. And also from the metagenomic sequencing and genome assembly, and we get some mechanistic understanding of the role of gut microbiome on intestinal barrier maturation in early preterm neonates. And that will help us for future development of rationally formulated live biotherapeutics. With that, I would like to acknowledge people involved in this project, especially Luke, Mike, and Lisa from Maryland Genomics that helped me to access the SQL2 program, early access SQL2 program, and also Shag and Powell at IGS and other people involved in the project, and my collaborators from pediatrics and University of Calgary and the funding agencies. With that, I will finish this talk. I will answer some questions later on. Thank you for your attention.
Thank you so much, Bing. That was a really terrific talk, and I see there's some questions that have come up, which we will get to at the end. Now I'm going to introduce our next speaker, George Weinstock. So for those of us who work in the field of genome sequencing, George Weinstock truly needs no introduction. Some of the highlights of his career include working on the Human Genome Project as co-director of the HGSC at the Baylor College of Medicine, and then as the associate director at the Genome Institute at WashU. So George has not only worked on the human genome, though, he has also led efforts to create the first assemblies of the rat, mouse, macaque, cow, honeybee, Drosophila, and sea urchin genomes. And sea urchin genome project is actually how he and I first met. George has never met a genome he didn't like, but he assures me that the sea urchin was absolutely his favorite. George then went on to become the leader of the Human Microbiome Project, and it's his work in this area that he's going to talk to us about today. He's now at Jackson Lab, and you may not know this, but Jax is now a testing site for the coronavirus, and so we're very happy that he's able to take time out to join us today. So I'll pass it to you now, George.
Thanks, everybody, for taking some time out of your stay-at-home day. I'm going to talk about some of the things I'm going to talk about are touched on by Bing already. I'm going to focus more on using 16S reads to learn a lot more about strains and microbiome than you might have thought you could do. So one of the issues has to do with phylogenetic placement, and I'll talk about what the challenges are with the different types of sequencing for that. But the other one has to do with looking at polymorphisms in 16S gene sequences that can discriminate different strains. So we get down below the species level to the strain level. The importance of that, as you'll see, is that strains can have functional differences, and sometimes those functional differences, which are encoded in the rest of the genome, that haplotype is very tightly linked to the polymorphisms in 16S genes. So by just looking at the 16S gene sequences, you can infer something functionally about the strains if they'd previously been studied. So in terms of phylogenetic placement, this is sort of one of the big issues in microbiome studies. We're continuously discovering new organisms that haven't been described before, and where do you place them if you just have a 16S gene? So the top paper that's referred to there, Novel Bacterial Taxa in the Human Microbiome, was a project that my group did with Katie Pollard's group in San Francisco during HMP1. And we were trying to ask the question then, how many new organisms not previously described are in the human microbiome? Since this was an HMP1, this was the first really large human microbiome data set that we had to play with. And we really had to stand on our heads to answer that question, because using the 454 16S sequencing that was done then, there's a lot of chimeric sequences, a lot of other artifactual sequences. So when you find things that are not in databases, it turned out that overwhelmingly meaning like greater than 90%, maybe 99%, were some kind of artifact. And so we had to use a combination of short reads from Illumina and medium-sized reads from 454 in order to answer the question, how many of those sequences are for real? And as you'll see later on, this is actually much easier to do nowadays than we had to do it then. The answer, by the way, is because there's been so much work on the human microbiome that there's relatively few new sequences that we run across at the level of sampling that we do for most microbiome experiments now. There's still some, but relatively few. Of course, now that I'm at Jackson Laboratory, we've focused a lot on the mouse microbiome, which one of the surprises for me was actually the microbes in the mouse microbiome, while functionally related to what are in other mammals, are in terms of the actual species that are there, many of them are not in databases and they hadn't been described before. So there's a real effort there at dealing with the what are called the unclassified sequences, because you can't find them in databases, or if they're in a database, there's just somebody saw it once before, but there's very little information about the organism, and usually they haven't been phylogenetically classified. So this becomes a huge challenge as we do more and more microbiome studies to complete our catalog of what the organisms are that are present. So here's an example of the second topic about getting some kind of functional information by looking at microbiome sequences, 16S sequences. This was a project also part of the HMP1 era that I did with Huiying Li, who's a professor at UCLA. And in that case, we were going to study Propionibacteria acnes and its relation to acne. A lot of the bacteria on your skin are P. acnes, but there are different strains. And so we wanted to test the hypothesis that some of the strains would be pathogenic, some of the strains would be just neutral, commensal, and so forth, and try to do that. So we isolated samples from lots of people, both from acne lesions as well as from healthy skin. And then we did full-length sequencing because we didn't want to have to do shotgun sequencing in those days of all of the genomes of all of these many hundreds of acne strains that we were isolating. So we decided we would take the leap and use the 16S sequences, what was called ribotyping in those days, in order to classify the different strains of P. acne. And ribotyping had been around for a while, particularly with pathogens where you're trying to identify if you see a particular species, is this going to be a highly virulent one or not? And it turns out that there's a number of pathogens where there's a correlation of their virulence to the polymorphisms you see in their 16S genes, their ribotyping. So that's what we were going to apply to this. So here's an example of that. So we did full-length sequences on 16S, and in those days, the way you did full-length sequences was Sanger sequencing. And Sanger sequencing, even though it gave you a 750 base read or so, you had to clone the gene. You had to then sequence in from both ends and usually do a third read in the middle in order to get a high quality full-length sequence of that particular 16S gene that you had cloned. So we did this for over 30,000 of these, and then took the top 10 in terms of abundance ribotypes that we found. So in the second column of the chart, you can see here's the difference between the 16S sequences we're seeing. It's usually only 1 or 2 nucleotides different because these are all the same species. And this is the frequency of polymorphisms that you see with P. acnes. And you can also see the number in that second column is the position in the 16S gene. And so you can see they're all over the place in the 16S gene. There's not a particular hotspot, a particular variable region. Anyway, based on that, you'll see here are 5 different ribotypes that are found almost exclusively in acne lesions. They're not found very often in the normal skin of people. And here is a ribotype that is found almost exclusively in healthy skin, hardly ever in the acne part, as if it might be protective or highly beneficial. And the other ribotypes that are there you see are sort of 50-50 between the lesions and the healthy types, so they're not really contributing anything. So this is the kind of thing that gives you reason to believe that looking at 16S polymorphisms and using that as a way of finding strains can then have good information for functional consequences. And we did the whole gene, we isolated a number of these bacteria, did whole genome sequencing of them. And these are clustering, these are all whole genome sequences here, and they're being clustered. And the main point here is that those 16S polymorphisms were reflected in the whole genome sequence. They clustered together according to the ribotypes, and that you can see there are differences both in terms of plasmids as well as in terms of other parts of their genome. And this is now, you're getting down to the level of really being able to do functional analysis, look for what genes distinguish a protective one from a pathogenic one from a neutral one, and so forth. And so Huiying has followed up a lot on this and has published some papers having to do with porphyrin metabolism, things like that, that are some of the distinguishing things that seem to be related to pathogenesis. Okay, so in work that we've done more recently, and this was a project in my lab where now we could use PacBio sequencing. This was led by Jethro Johnson, who was a postdoc, and he's now at the University of Oxford at a microbiome center at the Kennedy Institute there. Here, we were interested in trying to show that the quality you could get from what now is called HiFi sequencing, but then just the idea that you could go around the circle many times and get a consensus sequence that was high quality, was that good enough to be able to pick up these kinds of polymorphisms in the genome? And a lot of this work was done back in the RS2 days. We're doing more with Sequel 2 now, but with a slowdown in the reagents because of the coronavirus thing. It'll take us a little while to get through all that we want to do there. So I'll show you some of the RS2 data. So in this case, we're looking at E. coli. So in the upper left, so E. coli has 7 16S ribosomal RNA genes, and they have known sequences and known polymorphisms. And that's what's actually shown in the upper right for E. coli K12, the polymorphisms that you detect from the sequence. And their relative frequency has to do with the number of those 16S genes they're in. Some of them are only in one of the 7 genes. Some of them are in all of them or in just 6 of them and so forth. So in panel A in the upper left, we made a mock community of 36 strains that had E. coli K12 in it. And we did full-length 16S sequencing from that, and then compared those sequences to an E. coli reference sequence, and we were able to detect those polymorphisms that you see there. And they're very similar, almost identical to the polymorphisms we see in the in silico analysis of the E. coli to its right. Then underneath that is a pure isolate of E. coli that we did shotgun sequencing on the MiSeq, and then identified polymorphisms in the 16S gene from that. And you see they're almost identical to the ones that we see either in the in silico one or in the mock community PacBio long read one. Shown down at the bottom in all the colors, there's 4 colors there for the 4 bases are the 7 ribosomal RNA operons, and these are 4 of the regions where you can see polymorphisms, and it shows you exactly what kind of variation you see there, what the base changes are. And then in terms of the utility of this, in the lower right is the E. coli 0157 sockeye, which is, this is the highly pathogenic E. coli strain that has a slightly different polymorphism set than E. coli K12 is. And here we're showing that you can distinguish these 2 E. colis apart from each other, even though there's only a few mutations by looking at these polymorphisms, completely analogous to what I was showing for the P. acnes strain, except this is now applied to E. coli. So, okay, so all of this encouraged us that putting effort into doing full-length 16S sequences had a lot to offer in terms of what we could learn, both about the microbiome, about pathogens, and so forth. So here were our options that you can do. You could do Illumina short read sequencing for 16S, and that will only give you partial sequences, which I'll have more to say about in a moment. Or to get the full length sequences, already talked about the effort that's required to do Sanger sequencing, PacBio sequencing. We're all here today because we know what's involved with that. You could use the short range sequences from Illumina and use one of those platforms like 10x, or there's some other ones where you sequester a long molecule, make it into short reads, do the short read sequencing, and then put it back together. So in principle, you could get a full-length 16S sequence from that. Not sure if anybody's done that yet. And then with Oxford Nanopore, its read length is certainly long enough to be able to do full-length 16S sequencing, and we've done a certain amount of that, but we cannot yet quite get the accuracy that you could get with Sanger or PacBio in order to be able to confidently call polymorphisms versus sequencing errors. So we tend to favor the PacBio method for full-length sequencing. Now, if we compare full-length sequencing to the sequencing just parts of the 16S gene, so at the top of this slide is the 16S gene and showing regions of high and low variability throughout it, the reasons why they're called variable regions. And then at the bottom of that figure are the various partial 16S regions that have been sequenced either back in the 454 days where HMP1 did the V3, V5 region almost exclusively, although did some V1, V3. Now with Illumina, we tend to always do V1, V3 using the paired 300 base reads. But other people, as you heard in Bing's talk, use V1, V2, V4, some of the other partial regions that are available to do things with. Now, what's the difference between those? So we did an experiment where we went into the database of 16S genes called green genes, and we built a sub-database out of high-quality full-length sequences that we got out of there. We got about 14,000 sequences altogether, and they were non-redundant. That was the other characteristic. And so we then took those full-length sequences and we pared them down so we would either have a V1, V3 set of sequences, a V3, V5 set of sequences, a V4 set of sequences, and so forth that came from that same database. And then we compared using methods like the RDP classifier, how accurately you could get the right answer for what each of those sequences was by comparing it back to the same database that it came from. So in B here, we're talking about how many sequences, what fraction of them could you not identify accurately to the species level? And so you see in the middle of that histogram is V1, V9, that's the full length sequence. Almost all of those could be accurately identified to the species level. And then you can see V4 there where more than half of them could not accurately be identified at the species level. So you're sort of locked into the genus level or higher for things like that. And then there's varying degrees of the other sequences. You can see the best of the partial sequences was V1, V3 there. And then to the right of that are these trees. So this is, we took all those 14,000 sequences and built a tree. And so and then for each of the branches of the tree, we've colored it as to where the greatest inaccuracies are. So the more red it is, the more inaccurate it is. So you can see with V4, they're just about all of the different taxonomic clades have difficulties, have problems. With the V1, V9, the full length sequences, we don't really see any issues. If we take V3, V5, what we used for the Human Microbiome Project, you can see in that one down around 4 o'clock, there's a clade. Those are actinobacteria that are probably the least accurate. For the V1, V2 region, you can see right up at the top around 12 o'clock, that's the proteobacteria, that's the least accurate region, and so forth. So you can definitely get the idea that these partial sequences are not all the same in terms of the kinds of biases that they give you, and that the full length sequence is certainly definitely the gold standard, no question about it, showing by this. And then in D, for those of you who are OTU producers, we took each of these sets of sequences and ran it through a sort of standard OTU production pipeline to ask how many OTUs would we get given that there should be 14,000 sequences. And so for the full length one, we got almost the right number of OTUs. And again, for V4, we got the fewest number of OTUs in terms of what was being produced there. So this was our attempt to be rigorous and show that it really is worth putting the effort into full length sequences. Here's what you get over using the other methods that are there. And if it's valuable to you, this gives you a way to do the cost benefit analysis. The accuracy issue. So here is how many cycles should you do? taking these sequences, aligning them. This is from the mock community, taking different numbers of cycles in the PacBio sequencing and aligning it to the 36 genomes that were in our mock community, and then getting error rates. You can see on the right that the substitution error rate is sort of independent of the number of cycles. That's because those are not actually errors. Those are actually the polymorphisms that we're seeing. The error rate is actually much lower than that, so you don't really see it. And that's another argument that the sequence accuracy is good enough to be able to see the polymorphisms. You can see, however, that for indels in the two middle slides, it's when you get to around 10 cycles, now you've probably done as good as you're going to get in terms of indel error rate and everything. So we sort of have geared most of our sequencing with, if we're going to filter things and go for the higher quality, we're going to do the cutoff around 10 cycles. All right, now here's another, now we're getting into some real microbiome data. In this case, these are stool samples. So we have in the upper left an A, these are stool samples from 4 healthy individuals. These are downhill mountain bike racers, they're athletes, and the names of the samples are named after their bicycles. And when we do the standard microbiome analysis, 16S microbiome analysis stool samples, here we're just going to look at Bacteroides now, because that's one of the dominant organisms in the gut microbiome. You can see that Scott and Iron Horse were sort of high Bacteroides, and Commensal and Breezer were lower Bacteroides. In the people who do enterotypes, these probably would have separated into 2 different enterotypes by those definitions. Now, the y-axis is the frequency of Bacteroides. This is at the genus level, doing full-length sequencing. The x-axis is the frequency of Bacteroides doing V1, V3 sequencing. And it just shows you that you get the same abundance at the genus level for Bacteroides for these. But now if we look at B underneath this, and we use the full-length sequences, or we try to get down to the species level, down on the x-axis, you can see there are 9 different species of Bacteroides that we detect in these samples. And we've done 3 different types of sequencing. We had the V1, V3, and the full-length 16S that we were in panel A, but we also have here shotgun data, MySeq shotgun data, to look at these different organisms. So what you can see is, even though at the genus level, there's a certain frequency of Bacteroides for all 4 of these samples, the particular species that each of the samples has is quite different from each other. And therefore, just to say that you were going to do some statistical analysis based on their Bacteroides, you were going to miss a lot of important information because the species are quite different. In general, we can detect species with V1, V3, and V9. However, you'll notice the 5th one in from the left on the x-axis, Bacteroides intestinalis. If you look through there, you'll see that you're only able to detect that with the shotgun sequencing or the full-length sequencing. You can't detect that with V1, V3. The critical regions for defining that organism are just not in the V1, V3 region. So this is another theme of this. Doing full-length sequences gives you this is one of the reasons why it gives you a greater definition, because sometimes the important sequences are outside of the region that a partial sequence is giving you. Okay, let's skip the stuff on the right and go on to the next one. So here is now again looking at our mock communities, and here we can see again we're comparing V1, V3 versus full length. There's 3 species down at the bottom, a bifido, a gardnerella, and a prevotella, that are picked up by full-length sequencing, but are essentially completely missed by the V1, V3 sequencing. And there's another bifido strain, bifido longan, that is measured to be a much higher abundance with full-length sequencing than it is with the V1, V3 abundance. Again, bifido seems to be one that's not picked up very well by partial sequencing. So There's always a lot of discussion about when you do OTUs or when you do these definitions of taxa, what percent identity for everything from 100% identity on. We tend to do all of the above every time we do an analysis because the different types tell us different things. So here I'm showing you if we do a 99% clustering or a 97% clustering, and again, we're comparing V1, V3 partial to full length. You can see the full length does much better in terms of giving us the most matches to species in the databases, whereas with the shorter sequences, they either give you an ambiguous answer because they match multiple things, or they just get the wrong match because they're too short. Because PacBio can sequence much longer than a 1500 base pair sequence, our friends at Shoreline Biome have done what they call extended sequencing, where right next to the 16S gene, you'll see here in the purple, the internally transcribed space or the ITS sequence before you get to 23S. And so they've developed some amplicon sequencing with PacBio that not only does the full 16S gene, but includes also ITS sequencing and amplifies part of the 23S gene, which is where the consonant region is for the other primer. And what this does is it gives you another region with more variation than in the variable regions of 16S. As you saw for the E. coli and for the P. acnes, we're really dealing with just a few polymorphisms. And whereas the accuracy of the sequencing is good enough to see those, if you now include the ITS sequencing, you now get a much greater number of variants. And so you're in a much better position to be able to do the discrimination. The downside of it is that there aren't quite as huge databases about ITS polymorphisms yet as there are for just the 16S. And in addition, the 23S gene can have a variable length. And so there are some well-known organisms that don't amplify well by this method because they have a much longer region that they have to amplify. And so you get a bias from that. But nevertheless, definitely worth keeping in mind that for a particular organism that you're going to study, and you can generate your own sets of ITS data and that are correlating with different strains, now you have a good way of doing that kind of phenotype-genotype relationship where the genotype is going to be the ribosomal RNA regions and the phenotype is going to be virulence or acne or whatever it is that you're looking at. So just to wrap it up then, I think we're making progress. We have a daunting problem because there are so many organisms in the human microbiome and the mouse microbiome that we have to do. We have to expand databases, correlate with phenotypes, do all of those things. And right now, it's still a little bit costly. We'd like to see the cost come down, and we'd also like to see higher throughputs so that what we can do with long read sequencing becomes just as cost effective as what we can do with the high pooling that we do with Illumina short read sequencing. So I will stop there. We have lots of projects and lots of collaborators where we've applied this with. I'd like to give a special shout out to Jethro Johnson, though, who really took the lead on moving the full length 16S with PacBio project along. Thank you.
Thanks, George. That was really informative. And I hope everyone else enjoyed that as much as I did. So we now have some time for some questions, and I can see that there's quite a few that have populated the question screen. So the first one is, how well does PacBio sequencing identify copy number of ribosomal RNA within species, including when performing assembly? George, maybe you can talk about that.
Okay, well, there's a type of polymorphisms that we detect where there's actually the same polymorphisms in different strains, but there are different numbers of the 16S genes in one strain that has a polymorphism compared to the other. And so here we're relying on distinguishing the strains, not on the locations of the polymorphisms, but on the within genome abundance of those polymorphisms. And so we can get quite accurate using that. And you saw in the E. coli where we had 7 16S genes that we could distinguish those. Now, in terms of just counting 16S genes, we've had good luck in doing assemblies with long reads. We've done both PacBio and we've also done Oxford Nanopore. We have one strain that has 17 16S genes, and we could separate them all in the assembly of a long read. So I'm not sure if that exactly answers the question that was being asked, but it's sort of a new day now in terms of being able to accurately describe 16S genes in a complete genome.
Awesome. Okay, so let's see, here's another question. Are there upcoming reference resources to map ribosomal RNA full operon amplicans covering from 16S through 23S? George, since you've used Shoreline Biome, maybe you can comment on that.
I think you should. That's a great question. Take a look at the Shoreline Biome website and their kit for the extended sequencing. They definitely have put together a database that they use for those things, and it's a growing database. I think this is a project that is in the works, and but I think that they are certainly open for people who are using that kind of technology to contribute to their database as well.
Awesome. Here's another one for George. Did you compare methylation patterns between non-pathogenic and pathogenic E. coli and the presence or absence of methylation-dependent endonucleases like MCR-A, MCR-B, MRR?
We haven't done a specific study of methylation. Back in our RS2 days, we did a lot of for a lot of these strains we were isolating. We did the methylation analysis that you could do with PacBio, and then that sort of quieted down for a while in the PacBio world. But we're very keen on doing more of that now that we're using Sequel II more and getting more whole genome sequences. But no, we haven't done a particular study that tried to look to see what's the relation of methylation and methylases to particular phenotypes.
Awesome. Yeah, and I'll just chime in also. We recently had a blog post on using the Sequel II platform to detect methylation. If you had any more questions about how that's working, the answer is it's working well. And you can read more about it at our blog. Let's see, I have a question for Bing. I saw there were multiple bifidobacteria assemblies in your PREMI data. Have you been able to identify metabolic differences between those species?
Yeah, that's a very good question. I'm still doing analysis, and so far we only get the complete genome for bifidobacteria rabies, and we're trying to get that for lungum and for other species. So now I don't have the answer for that because we're still doing the work, but that's certainly the things we're interested in doing.
Fantastic. So we have a couple questions about assembly. So perhaps we can have both of the speakers, and I'll chime in as well, comment on what software tools do you use to assemble shotgun metagenome data with PacBio? Bing, maybe you can start.
Sure. So I use Canoe. I think the version I use is 1.8, and for the long reads metagenomic assembly, it depends on how big the data set is. It might take a long time, but since at IGX we have this grid, so we can use a multi-node and run it, and usually can finish within like a couple of days. It depends on, again, it depends on how big the data set is. I think so far it works fine for us.
Awesome. George, do you do much metagenome assembly?
Not so much metagenomic assembly yet. I mean, we have done a lot of metagenomic assembly of short read data, right, but not so much with the long read data yet.
Right. Okay. So I'll also shout out, I see we have Alicia Klum on the line. Alicia Klum's a bioinformatician at JGI, who's one of our longest customers, actually. And she says that she uses Metafly out of Pavel Pesner's group at UCSD. And she says that it's fast and doesn't require error-corrected data, but you can provide it. Okay, then we have a couple questions about multiplexing on the PacBio. There's a question about how many samples can you multiplex on the Sequel II. So for 16S, you can multiplex 96 samples by following exactly the protocol that we've released or using the Shoreline Biome Kit. And then in terms of capacity, you can multiplex as many as 192 samples and still get at least 10,000 reads per sample doing that. And for shotgun sequencing, we have multiplexed as many as 8 samples, though I think 4 is or 3 is probably a more typical number for human metagenome samples like the gut microbiome. Let's see. Here's a question. I think this is for George. How do different factors like age and antibiotic, oh wait, no, I'm sorry, I think this is for Bing actually. How do the factors such as age and antibiotics usage affect the results? Do you have any data on that?
Yes, well, these are the important factors we examined in the context of IP and both in microbiota. We find age, or as a age in the form of gestational age, where gestational age is certainly affected in microbiota and IP. And we use analysis called NEAS analysis, or the balance tree analysis. We find out that actually the gestational age or postnatal age are the biggest factors, one of the biggest factors that affect microbiota. Is that all your questions were?
Yeah, I think that's a great answer.
Yes.
Yeah. Okay, so with that, that's all the time that we have for questions today. And so I want to end by first thanking Bing and George so much for sharing their time today and their expertise and perspectives in long-read sequencing. And then I'm going to mention 3 housekeeping things. First, we have recorded the webinar, and it'll be available for download in the next couple of days. You can watch out for a follow-up email with a link to the recording. Second, you can stay up to date with other upcoming webinars by visiting and bookmarking pacb.com/events. And finally, if you enjoyed this webinar, you might also enjoy our upcoming the upcoming April 2nd webinar by our partners, Shoreline Biome, where they're going to talk about how the combination of PacBio sequencing and their Shoreline Complete Strain ID companion software and curated full-length 16S database will let you comprehensively, rapidly, and inexpensively identify all members of the microbiome down to the strain level. And you can find the registration link on their website at shorelinebiome.com. And then finally, immediately following the webinar, you're going to get a very brief questionnaire. We would really appreciate it if you could take 2 minutes to fill that out, as it helps us develop new content for future webinars. So thanks again for joining us today, and I hope you'll join us again in a future webinar. And with that, be well, stay safe, and have a terrific day.